Hi, my name is Leda Guzman from Chile. It's a pleasure to be here. My presentation is about elucidating the death mechanism induced in gastric cancer cell line by triterpene from Quillaja saponaria molina. Gastric cancer is the most common cancer impacting the world population. It is the fourth most common type of cancer. It has been the cause of 77,000 deaths around the world during 2020. In our country, Chile, it is also among the most common type of cancer, although the incidence of gastric cancer has gradually decreased over the last 50 years. This disease continues to be an important cause of death in our country. There are two types of cancer, of adenocarcinoma, the intestinal and diffuse, but the intestinal is most common in the young men. The different risk factors are important in this pathology. For example, the infection with the Helicobacter pylori, the lifestyle, the diet, the low consume of vitamin C. Current therapy are based mostly on surgery, radiotherapy, and antiproliferative drugs. The most common antiproliferative drugs are epirubicin, cisplatin, paclitaxel, fluorouracil, fluoropyrimidine. Some drugs lack selectivity, impacting cancer cell and normal or healthy cell at the same time. In addition, some drugs have significant secondary effects. For example, nausea, vomit, diarrhea, hair loss, low blood count. Current drugs may be ineffective when patients develop resistance to the drug. Resistance has been observed in 50% of the patients. Therefore, new therapy based of new compounds are required. Quillaja saponaria or Quillaja III will be a new source of antiproliferative compound. Quillaja saponaria is a native to the central region of Chile. The infusion of this bark has been traditionally used as soap, shampoo, as well as medicine by indigenous people for centuries. Quillaja extracts are used in wide range of applications. For example, as a surfactant for food and cosmetic industry, and the source of vaccine adjuvant. The property of Quillaja extract are based of triterpene compound. The most abundant type of triterpene Quillaja are saponin, a family of more than 50 glycosides of quillaic acid. One of these glycosides is QS21, is currently the focus of intense research because its uses as vaccine adjuvant. In this figure, you can see the quillaic acid is a hydrophobic compound. This quillaic acid is the aglycone. In the right side is a saponin. For example, QS21. Contain two hydrophilic tile. This structure is an amphipathic glycoside of skillatic acid because contain this hydrophilic tile. Previous research by our group has determined the selective antiproliferative effect of both compounds in gastric cancer cells during in vitro experiments. The aim of this work was to characterize the mechanism of skillatic acid and QS21 induced cell death of the two human gastric cancer cells has SNU1 and KATO III. Now, there are three separate stages may be identified during the cell death. A live cell is when both mitochondria and plasma membrane are intact. The vulnerable state is when the cell, which may belong to type 1, having damaged mitochondria and intact plasma membrane, or type 2, having damaged plasma membrane and intact mitochondria. And that cell, both organelle, both mitochondria and plasma membrane are damaged. Two basic types of mechanisms involving in the cell death. The sequential mechanism is when the alive cell passes to vulnerable cell and then dead cell. Non-sequential is the alive cell pass quickly and the dead cell. Or big question, which type of mechanism induce skillatic acid and QS21 in both types of the cell? Our strategy is assess the impact of increasing doses of skillatic acid and QS21 on tumor cell grow in vitro to reveal the stage of the cell death. We use fluorescent probes and detected by flow cytometry. Probe has propylene iodide, rhodamine, HERT, or antibody, or annexin 5. The cell was deprived for 24 hours, are synchronized, and then are incubated with the QS21 and skillatic acid, or staurosporin has a control. Then, or after 24 hours, the cells are stained with the probe. In this figure, you can see the four stages. Alive, when the mitochondria is intact, the fluorescent is green. When the rhodamine binds to functional mitochondria. The cell, the dead cell, is when the plasma membrane is broken and the fluorescent is dead because the propidium iodide is bind to DNA. There are two vulnerable states. Vulnerable type 1, when the one parameter is broken, for example, mitochondria, or vulnerable type 2, when the membrane is broken. Experiment at increasing doses of quinine acid on QS21 allow to determine the damage sequential index. It's a quantitative criterium to discriminate between both death mechanisms, sequential mechanisms and non-sequential mechanisms. To determine this index, the doses of the compound leading to 50% of both alive and dead cells must be determined from plotted data. The relation between AC50 alive and AC50 dead. You can see the percentage of cell viability and the different concentration of the compound. The sequential mechanism, there is a big difference between the live cell and dead cell. And you can see the vulnerable cell. In this case, when the non-sequential mechanism, the vulnerable cell is very slow. And then the AC50 alive is very similar with the AC50 dead. It's very close. Now, the region between AC dead and AC50 alive permit to difference between both mechanisms. When the index is higher than 1.1, the mechanism is sequential. If the index is smaller than 1, it's not sequential. The result of this experiment, of our experiment, doses response data support of quinine acid induced sequential effect on KATO III and SNU1 cell. QS21 induced no sequential effect on SNU1 cell. This figure shows that the described in the previous slide. You can see the sequential mechanism, and in this figure it's not sequential mechanism because it's very close the alive and dead cell. In this table, in this column, you can see the index, and QS21 on SNU1 is 1. In other case, the index is the number is over 1.1. This figure shows the sequential mechanism because the cell is alive, then to pass a vulnerable state, and then death. 3 states: alive, vulnerable, and death. In this case, the alive cell passed quickly to death. The QS21, the effect on SNU1 is very stronger. It's very strong. In order to confirm this study, TUNEL assay were realized. The SNU and KATO III were incubated with the compound and 2 concentration, QS21 10 micromolar and Kijai acid 100 micromolar. The white arrow show the DNA fragmentation in yellow color, and in this case is the pink color. This figure shows the plot. The TUNEL, the percentage of TUNEL positive with the SNU cell and the KATO III cell. You can see when the cells were incubated with the QS21, the percentage of a TUNEL positive cell was the 82%. In KATO III, the percentage was the 50%. Now, the difference is not similar with the effect, the Kijai acid. The effect is more important with the QS21. Another, the SNU1 is more sensitive than Kijai acid. Excuse me. Is more sensitive than KATO III. Another assay was analyzed the apoptosis using annexin 5 and propion iodide. Both compounds induce apoptosis on SNU1 and KATO III cells. You can see in this figure the cell untreated, the positive control with cisplatin, and both compounds and the similar concentration. Kijai acid 100 micromolar and QS21 10 micromolar. You can see the cell in apoptosis, late and early. Furthermore, or in order to confirm this result obtained by TUNEL assay, we employ flow cytometry using caspase 3 and 7 activity, and 2 compounds were or induced the caspase activity compared with the cell untreated. Now, with this result, we realized a molecular docking with several cancer-related proteins. The table shows the free energy in kilocalorie per mole and other physicochemical parameters. You can see the different energy, free energy, with the different protein. The most important result was with the bit protein. The bit protein is a pro-apoptotic protein. The free energy was minus 8.2 to Quest 2021 and minus 9.9 with Kijai acid. This figure shows the protein, the pocket, the protein, the interaction with the different amino acids with the both compound. The more important amino acids and the more important hydrogen bonding and hydrophobic contact. For example, Quest 21 are important glycine in position 43, aspartic, or glutamic. And the hydrophobic contact, the important phenylalanine and tryptophan. Finally, we analyze the mitochondria using immunofluorescence by flow cytometry, using the antibody, again, the cytochrome C. Kijai acid induces the liberation of the cytochrome C on KATO III and KATO III, in lesser degree than Quest 21, but no induce the liberation of cytochrome C on SNU1. You can see the SNU1 Kijai acid induce no liberation of cytochrome C in chain. KATO III induce the cytochrome C, but a Quest 21 induced the liberation the cytochrome C in both types of cell, KATO III and SNU1. As conclusion of this work is Kilaha saponin induced apoptotic death of human gastric cell in vitro. The activity is selected because the both compound induce a different way or different mechanism, non-sequential or sequential mechanism. Both compounds induce that using different way and co-interact with the cell through different targets, maybe the receptor or bit protein, perhaps use a channel in the membrane. The question is possible to answer in the future. Now, acknowledging, I want to thank the whole group, the researcher, Leandro Padilla, Alejandro Corbalán, Juan Carlos Gentina, Juliano Bernal, and other students has